myself Halavat Ramesh. Especially today we will talk about very general topic, especially related to biological subject. Especially today my topic is biological activities like MTT Assays, cytotoxicity, antioxidant activity are some clinical research methods. And this is the some basic topics in biological activities. Today we will discuss like basic introduction parts, the principles and what is methods we will discuss today, so slide by slide. You can see the first one, biological activities. Biological activities is the word, is the capacity to specify the molecular entity to achieve a defined chemical effect. The target is the measurement of activity or the concentration of molecule required to cause the activities. Biological activities always measured by the biological assays. Simple, you can see what is biological activity means the capacity of the specific molecules entity to achieve define the biological effect. That is the main point of the biological activity. You can see next one is the what is determination of biological activity. How we will determine. So biological activity is the molecular entity is defined as the A is equal to CF. Suppose you can see A is equal to CF. A is the activity, C is the amount of substance concentration, and F is the parameter designed as inherent activity. Then you can see what is that biological activity of protein. So this is, you can see the biological activity of protein, immediate and of various protein functions. Suppose the protein in good condition, it will be work on these functions. You can understand the concept that only the protein have the activity. It can perform its function. As the protein is biological activity is called active proteins. Namely, the active protein has the biological activities. So active proteins are shows the biological activity and their functions. You can see next one slide. What is the basic unit of biological activity? You can think the cell is the one of the smallest unit living things. A living things either made up of one cells like bacteria or like any other human cells are called organism. The cells are the basic building blocks of all organism. So one cell is the one of the mutation then will form to all organism. So biological activity occurs as the result of certain effect from exposed to the molecules. These effects a metabolic or a physiological response. Biological activity is defined as being applied to the simplest and the most complex reaction and molecular system. These are many sort of biological activities. These activities can be studied in vivo and in vitro. So the biological activity always depends on the dose given in the living organism. So it is the biological to show either the beneficial or adverse effect that range from low to high. Absorption, distribution, metabolism and excretion are the main action used to measure biological activities. You can see next one. Biological activity is also called as the pharmacological activity. Simply you can say biological activity means nothing but a pharmacological activity. It describes the benefits and adverse effect of the organ or the drug on living matter. The activity depends on the ADME, absorption, distribution, metabolism and excretion. So when the drug is complex or the biological activity exerted by the substance active ingredient or chromophores. Then you can see what is when you talk about the biological activity, the familiar word is in vivo and in vitro. What is in vivo? The happening or existing inside the living body. So in vivo things is the living organism. Nothing but of in vivo is the living organism. The experiment will be carried out in the living organism. Next one, see what is in vitro. It performs or taking place in the test tube. Either you can do the experiment in test tube or either you can do in any glass or cultured dish and elsewhere outside or living organism. In vitro means nothing but of in a glass such as the test tube or petri dish plate. That is called in vitro experiment. In vivo means in the living organism like rats, frogs. These you will do the in vitro. In vivo in the test tubes, glass, or petri dish plates, etc. You can see next one. What are the main biological activities? You can see the biological activities. You can see many things. If you synthesize one compound, you can see the antibacterial activity, anti-tumor activity, antifungal activity, antiviral activities, and so many biological activities you can do it. The diabetics and the anti-cancer, antioxidant, hyperglycemia and hyperlipidomic effect. These are the biological activities. You can see the flow chart. You can see here the some of the biological activities. You can see anti-inflammatory activity, antimicrobial activity, anti-cancer activity, and antiviral activity, and the anti-biofilm activity, immunomodulatory activities, cholesterol lowering activity, antioxidant activity. These kind of the biological activities you can do it if you synthesize any molecules and any drugs. And you can see that few of the other biological activities, phytotoxicity, like if you isolated or extracted one of the phytochemicals from plant, either it will be shows the biological activity or that kind of activity you can do it, the phytotoxicity. And the anti-inflammatory, antifungal, anti-cancer, anti-HIV activity, and photocleavage activity, and the antibacterial activity and the allergical activity. You can see some of the other sea cucumbers, saponins, what are the activities you can do it. And you can see the citrus flavonoids, you can see biological activity, antioxidant, cytoprotective, anti-inflammatory, anti-photoaging, wound healings, anti-melanoma. These all are the biological activity. You can do it with any new compounds you isolated, or you can synthesize from any other source, or you can synthesize the drugs or something. You can see that first one is you can talk about the brief introduction about the MTT assays. Like MTT means medical training therapy. The MTT assay is the calorimetric assay for assessing the cell metabolic activity. So MTT assay is used to measure the cellular metabolic activity as an indicator of a cell viability, proliferations and cytotoxicity. The product name is the MTT assay is like a kit. Cell proliferation means detect method is calorimetric, and the sample type is adherent cells and suspension cells, and the assay type is quantitative, and the assay time is 3 hours 15 minutes. And you can see species activity is like react with mammalian and other species. So product overview is the MTT assay kits in easy to use and non-radioactive, and the through use for the measuring of like cell viability and cytotoxicity and proliferation. And you can see the MTT assay protocol summary. You can see replace the serum containing media with serum free media and MTT reagent in cell in culture, and incubate the 3 hours 37 degrees Celsius, and add MTT solvent and indicator for 15 minutes, and analyze the microplates in reader. You can see here the image like how you will see MTT assays. You can flat like and MTT assay is the multiple viable cells. You can do these kind of graphs also. And you can see this is the OD like absorption OD in 390 nanometer. MTT cells proliferation assays, you can see cell numbers, and you can draw the graph how it will be shows that MTT assay you can easily identify. Next you can see the another one method is cytotoxicity assay. This is also very good method to how that compound will shows toxicity. You can measure from this cytotoxicity assays measure the ability to cytotoxic compound to cause cell damage or cell death. So cytotoxicity assay is widely used in fundamental research and drug discovery to screen libraries for toxic compounds. In drug discovery, cytotoxicity is a critical endpoint when a evaluation the fate and effect of compound. The compound generating a cytotoxicity response may be eliminated from subsequent screening round when evaluating potential pharmaceutical treatment or a compound target rapidly dividing cell may constitute a hit in a drug screen aimed at identifying cancer therapy. So this assay can be classified according to measurement type of endpoint, color change, fluorescence and luminescence, etc. Okay. So cytotoxicity assays is classified in mainly four methods like dye exclusion, colorimetric assay, fluorometric assay and luminimetric assays. You can see here the dye exclusions you can use this Congo red and the Aristocins B assays and colorimetric MTT assays. You can use here crystal violet assay, fluorimetric and luminescence assay also. You can see here the what is dye exclusion assay. So variable cells exclude dyes, but is dead cells not excluded them. So determination of membrane integrity is possible. The variety of such dyes have been employed by the Notions, Congo red, Brosib B and Trifton use. Though comparatively is very simple and require a small number of cells and rapidly. This is one of the dye exclusion assay for the finding of cytotoxicity method. And you can see this is the dye exclusion assay, how the Trifton blue assay. You can see the live cell process intact with the cell membranes that exclude this dye, whereas dead cells do not. Cell suspension is mixed with the dye and then usually examined. Variable cells with having a clear tryptophan, whereas dead cells will have a blue cytoplasm. Counting is generally done using the hemocytometer. Next one, you can see the calorimetric assays. The reagent used to calorimetric assay to develop the color into the response of the viability of cell and they evaluate the metabolic activity of cell. So this is applicable for the suspension cell lines and inherent. This assay to platform to comparable economical also. MTT assay, this is you can find out here MTT assays methods and how it will be looks. And you can see calorimetric assays also is one of the method to identify or determine the cytotoxicity. You can see here next one also you can see LDH assay and next one fluorimetric assay is one of the method to do the cytotoxicity experiment. You can see next one, this is that fluorescence. You can find out fluorescent in like you can shown here the figure. You can see next one luminometric assays, the fast and simple determination of cell proliferation and cytotoxicity of mammalian cells, produce peristate and the stable grow and type signal after reagent is added. ATP assay simply you can see called this is used for cytotoxicity. Drastically it will be changed in ATP. You can see this kind of ATP. Next one is the another one topic is the antioxidant. What is antioxidant? You can find out and like free radical experiment you can do it. You can see what is antioxidant of protein and you can see what is free radical and aging. You can find out here also. And you can see how antioxidant electron protons transformation, how the radical is formed, stopping damage. Next one, clinical research. One of the clinical research trials is it will be like a in vivo studies. You can search for study of the test in new medical treatment for new way to using the existing treatment for the see if is the better way to prevent the screen the diagnostic and treatment disease. This is you can see here the preclinical phase 1, phase 2, phase 3 and ethical committee animals they also allow to. And characteristics of a clinical trials, you can see these are the characters we need to develop and you can see this is what is clinical trial. Thank you. Bye.